All right, welcome to part three, where I will address the biochemical evidence that is used to support evolution. First on the list of evidence is junk DNA, or the DNA that was supposedly inherited from our ancestors. To start, DNA is made of exons, which code for proteins and comprises about 2% of our genome, and introns, which do not code for proteins and comprise about 98% of our genome. So you may have heard that since 98% of our DNA doesn't code for proteins, that it was junk that was inherited from our ancestors. It turns out that the introns, or junk DNA, actually do have a purpose. All introns are transcribed into RNA. However, they are not translated into proteins, hence why they were called junk. What are they transcribed to? There are many types of RNA. mRNA was mentioned earlier, but there is also transfer RNA and ribosomal RNA used in translation. There is microRNA, small nuclear RNA, just to name a few. And each of these types of RNA has a specific function. These functions generally are centered around gene regulation, which is controlling how the exons are expressed. So introns tend to control the timing of the exons, the length to which exons, or the level to which exons are expressed, and the length of time that they are expressed for. An example is microRNA, which sometimes binds to a strand of messenger RNA to decrease the rate at which it is translated. Another example is seen in the development of the human brain in the embryonic stages. There is a gene that controls the development of the cerebrum that is turned on for a longer time than it is turned on for in chimpanzees. And this is one of the reasons that humans are more intelligent than chimpanzees. In part 6 of these videos, I will talk about gene regulation in more detail. I cannot do anything more in these short videos than scratch the surface. Gene regulation is a highly complex process that, for the most part, is not understood. However, what is known about it suggests that the introns do play a large role. One could say that the exons are like the what, and the introns are the when, why, and where. The exons are like the actor on the stage, but the introns are like the play instructions telling him how to act. Or if the exons are like the alphabet, then the introns are what tell the exons how to write a book. Of course, it is not known what every single intron does, but certainly some of them have a purpose. I think it could be argued that most of them do have a purpose, even if we don't know what it is yet, because of RNA's reactivity. That is, if an intron didn't have a purpose, then whatever RNA it is transcribed to would likely react in ways that are harmful to the organism. To give some more examples, junk DNA, or introns, plays a role in determining differences between cells in the same body, organisms in the same species, and species in the same organism or ecosystem. Each cell in our body has exactly the same genome. What makes, for example, a muscle cell different from a neuron is the timing, levels, and lengths of expression of the different parts of that genome. For this reason, it is likely that junk DNA plays a role in embryonic development, where a fetus develops from a single cell. The same concept is true of organisms in the same species. Using humans as an example, you and I have about 99.9% similar DNA sequences. What makes you and me different is largely the timing, level, and length of expression of the different parts of our genome. The same is true of species. Humans and chimps have about 98% similar genomes, but it is largely the timing, level, and length of expression of the different parts of our genome that makes us different, and this is at least in part done by junk DNA. In fact, it is said that what separates complex organisms like humans from less complex organisms like bacteria is not their sequences of DNA as much as how those sequences are regulated. So in summary, junk DNA has a purpose that is unique to each cell, organism, and species. If it isn't actually junk, then one can't use the idea that it is junk to say it was passed down. Also know that this concept also applies to vestigial structures, although I do not talk about it here. Moving on, you may have heard that we have about 50% of our DNA in common with a banana. We also have about 99% similarity with a chimpanzee, 90% similarity with a cat, and 50% similarity with trees. The logic is that if our DNA sequences are so similar, then we must have a common ancestor. We could also say that they have a common creator, but that idea is avoided because it invokes the supernatural. If you want to see this talked about in more detail, see my faith versus science videos. But anyways, these percentages only refer to the exons, which accounts for only 2% of our genome. The other 99 or 98%, the introns, were not counted, probably because they were thought to be junk. But if one counts the introns, it brings the percent similarity between chimps and humans to about 95 to 96%. One might say, well, that's still pretty similar. Not really. A 4 to 5% difference in our genomes is huge. This accounts for about 135 million base pairs. This is huge, considering that even one mutation can have a large effect. Besides, we know from discussing the roles of introns that what makes an organism unique is not so much its sequence of DNA, but how that DNA is expressed. For this reason, saying that chimps are 96% human because of their genome is like looking at two books, let's say Great Expectations and A Tale of Two Cities, and saying, because they use the same words, they must have a similar message. This would be false. Doing this neglects the frequency with which different words are used, the ways they are used relative to each other, and the length of time in the book that each word is used for. Given these variables, one could say that a small change in the book's words, even if it is only 4 or 5%, could produce a very large change in the book's meaning, like we see with the two books mentioned. Likewise, looking only at an organism's genome and saying chimps and humans are 96% similar may seem like a small difference on the outside, but neglects the timing, levels of expression, and length of expression of those genes. By the same logic, I could say a banana is 50% human, which obviously is not true. It neglects these other variables, as mentioned earlier, but it is variables like these that are largely responsible for determining differences between cells in the same body, organisms in the same species, and yes, even different species. So, statistics like the one addressed here are very misleading. One might say, well, if one mutation can have such a large effect in these other variables, then all we have to do to go from chimp to human is change the genes, right? That would be like a shortcut. But remember, with the book, even though there might only be a 4 to 5% difference in the words being used, one can't change them one at a time. They must be changed together, or what is produced is not a functional book. Similarly, changing one base pair at a time in a chimp will result in the death of the chimp, which would stop evolution. So, because statistics like these ignore other variables and the limited number of functional combinations they have, they are very misleading. The next evidence of macroevolution is microevolution. Microevolution, evolutionists would say, is visible proof that macroevolution is happening around us. And to the assuming person, it makes intuitive sense. Just repeat our three steps over and over again until one has a new species. And microevolution does occur. We see it all around us when animals adapt to their environments. In fact, those who believe in Genesis must believe that a limited number of species were on the ark and eventually evolved to give us the millions of species we have today. However, assuming macroevolution from that is flawed reasoning. The first problem is that it is not safe to extrapolate millions of years of macroevolution from our limited observation of microevolution. Most popular instances of microevolution happen within a year, and we are using that experience to explain how all of life evolved over billions of years. The potential for error is high. This is like taking a pistol with a 5-inch barrel and pointing it at a target that is 60 miles away. To assume that one will hit the target simply because the pistol is pointed at it is unsafe because there are other factors that influence the path of the bullet, such as wind speed and direction, the shape of the bullet, and air temperature and humidity, to name a few. Theoretically, the bullet would hit the target, but that ignores that there might be other factors. So what does one do? Well, they would test to see if the extrapolation is safe. So test the pistol's accuracy at 10 yards, then 50 yards, then 100 yards, then a quarter mile, and then maybe a mile, and see if there are other factors that don't show up at a short range. In the same way, theoretically, microevolution will result in macroevolution if given enough time. But it is unsafe to assume that because there might be other factors present. So what does one do? They would test it in a controlled environment. So take a population and breed it over the course of one day, then 1 month, then 1 year, then 10 years, then 20 years, and see if microevolution behaves the same way in the long term as it does in the short term. Historically, when this is done, just like the pistol, there are other factors that come into play. This is why no one has performed a breeding experiment and started with dogs only to end with cats or chickens. It is also why the Miller-Urey experiment yielded only a couple of amino acids. It is simply bad science to assume that microevolution behaves over millions of years in the exact same way that we observe it over the course of 1 year, especially when the evidence suggests that it doesn't. So, extrapolations are not necessarily false, they just need to be tested to know that they are safe to make. This has not been done with microevolution. At this point, one might object, saying, well, we don't have millions of years to show that macroevolution is true. My answer is that we shouldn't need millions of years. All we need is a period of time that is significantly longer than most instances of microevolution. So 20 years should be plenty. If one observes other factors in that time, which we have, then how much more would we expect there to be other factors over the course of millions of years? Besides, we have the benefit of controlling the environment of what we're breeding in order to speed up evolution. It is simply unsafe and bad science to assume macroevolution from microevolution. Now, it is possible that in the objector's mind, evolution is supported by things like the fossil record and genomic evidence. So for them, there is no need to test this extrapolation. But that is circular reasoning. This is assuming that evolution is possible, and then using that to verify an extrapolation that allows for evolution to be possible. One first needs to test the extrapolation. To do otherwise is circular. The second problem with microevolution is that it is a misleading term because the evolution that is happening is not really that micro. Keep in mind the Charles Darwin quote. It turns out that there is no such thing as a small successive change, which means, according to Darwin, that evolution absolutely breaks down. He was correct. We now know the reason for this. The bigger a step is, the less achievable it is by random mutation. To illustrate this, let's look at one famous example of microevolution, which are Darwin's finches. He found these on the Galapagos Islands, and some had apparently evolved bigger, stronger beaks so they could eat a type of seed that the other finches couldn't. This example of microevolution has been used as virtual proof for macroevolution. But let's take a closer look at all the anatomical changes that need to happen for the finch to evolve. If the beak is changing size and the beak attaches to the skull, then the structure of the skull also needs to change to accommodate it. If the structure of the skull is changing, then the length of the optic nerve that receives information from the eye also needs to change. The tongue would have to increase its length to match the larger beak. Since the tongue is a muscle, it needs a host of intramuscular proteins, such as titin, actin, and myosin, that need to be upregulated to supply the new muscle. And a muscle without nerve innervation is pretty useless, so let's add developing nervous supply to the tongue. And if the brain isn't able to process the information that comes from the tongue, all these adaptations would be useless. So let's add new circuitry in the brain. And if the beak is getting larger, that will exert some new torque on the neck. So let's repeat the process for the muscles in the neck. and the list goes on and on. We know that all these changes need to happen simultaneously. For example, if the finch has a big beak but the skull hasn't been remodeled, that beak falls off, the bird dies, and evolution stops. Or if the skull is remodeled but the optic nerve hasn't adjusted its length, then your bird can't see, it dies, and evolution stops. All of them happen at once. If they don't, then evolution stops. So the small successive changes that are needed for evolution to happen do not exist. We can imagine this anatomically, but we will truly see it when we look at the biochemistry behind these small changes. So when one looks at a finch on the surface, it seems easy to grow beak size, like it wouldn't be that big of a change. But if it is looked at in more detail, like one would look at this slide under a microscope, one would see that there is much more going on beneath the surface than we were aware of. I will explain this in more detail later, but the point is that what is a small change in fitness is a huge change biochemically. I compare this to looking at two cars. We have a Honda Civic on the left and a Toyota Camry on the right, and saying, well, they look similar, so it must be easy to go from one to the other. Maybe there was a random error in the factory that produced the Civic, and out popped a Camry. And if one only looks at the outside, this is conceivable. All that is needed is some blue paint instead of white and a few rearrangements in the structure. But anyone who knows how a car actually works would say that there is a lot more that needs to happen underneath the surface. Examples include recalibrating the transmission, perhaps changing the engine type and reprogramming the computer chip, all complex processes. So, what seems plausible on the surface actually isn't, at, at least not by random chance, and so it is with microevolution. In Darwin's day, they had no knowledge of biochemistry. All they had was external anatomy to compare organisms by. So it is understandable if he was mistaken. But that is no longer the case. So remember the progression of skulls given in part 2? Or you can read Richard Dawkins' explanation of how the eye evolved and many other explanations like it. These take into account only anatomy, which makes it look like the steps are small. But they make the fatal mistake of ignoring the biochemistry, which shows just how great these jumps actually are. There is more to come on this later. Since the steps are so large, a random process like evolution cannot bridge them. Therefore, biochemistry should be the death of Darwinian evolution. By the way, I am approaching the subject of small changes from a biochemical perspective. It can also be approached from a mathematical perspective, yielding the same conclusion. By mathematically representing the chemical properties of what reacts in our bodies, it can be shown that the steps are not as small as we once thought they were, especially with more complex organisms. In turn, it is these same big steps that point to a designer. So if microevolution cannot occur without a designer, then how could macroevolution? Macroevolution would be a miracle. So one may ask, can we infer microevolution or macroevolution from microevolution? The answer is no. These large steps actually point us toward a designer, not away from one. They are too large to accomplish by random mutation. Evolution is, by definition, a random process, so this fact would debunk evolution as we know it. Yes, perhaps the designer made things so they could evolve on a large scale, but that would no longer be a question of science. So in summary of part 3, we address 3 pieces of biochemical evidence that evolutionists often give for their theory: junk DNA, DNA sequence similarities, and microevolution. We found that junk DNA is not actually junk, that it likely explains many things, such as the differences between cells, organisms, and species. For DNA sequence similarities, we found that species actually aren't that similar if we count the 98% of DNA that does not code for proteins. It also neglects epigenetic changes, which play a large role in differentiating species. And for microevolution, we discussed that it is an extrapolation that has not been experimentally verified. More so, microevolution actually entails many simultaneous changes, which according to Darwin, utterly destroys his theory. Darwin needed organisms to evolve in small steps because anything beyond that could not happen randomly. As was shown and as will be shown in part 4, those small steps do not exist. That is the end of part 3. In part 4, I will start the offensive evidence against evolution by introducing the topic of irreducible complexity. See you there.